are authorized to declare recesses of the committee at any time. Good morning and welcome to today's hearing entitled The Science of Zika, the DNA of an Epidemic. Let me recognize myself for 5 minutes for an opening statement. For more than a century, humans have been at war with the mosquito. In 1901, a U.S. Army physician named Walter Reed led a team in Cuba studying diseases that proved yellow fever was transmitted by mosquitoes. Since that discovery, governments, scientists, and individuals have pursued ways to prevent and control deadly mosquito-borne outbreaks. From yellow fever to dengue fever to Nile virus, humans are constantly under siege from new mosquito-transmitted diseases. Today, we will examine the state of science in the most recent battle in the war against mosquito-borne disease, the Zika virus. While for most people, Zika causes only mild illness, the Centers for Disease Control and Prevention have found that there is a link between Zika infection during a woman's pregnancy and severe birth defects. For some adults, there could also be serious neurological impacts. Summer is coming, and so are the mosquitoes that spread the Zika virus. The Gulf Coast of Texas, among other places, has been called ground zero for this type of mosquito that carries Zika. Zika infections have spread to over 80 countries and territories in Latin America and the Caribbean. We have seen local transmission of the virus in U.S. territories. In addition, over 500 people in the United States mainland have acquired the Zika virus while traveling out of the country. Over 300 of these are pregnant women. These dangers raise serious questions about the administration's handling of travel alerts. The CDC has issued Level 2 alerts for 49 countries and territories, which advise travelers only to, quote, practice enhanced precautions. They have not issued any Level 3 warnings to, quote, avoid non-essential travel, as they did during the Ebola epidemic in West Africa. The World Health Organization in February declared Zika a public health emergency of international concern. Such declaration is reserved for a situation that is, quote, serious, unusual, or unexpected, carries implications for public health beyond the affected state's national border, and may require immediate international action, end quote. Why has the administration not raised the travel alert level for countries with the highest number of Zika infections, such as Brazil and Colombia? Is the administration so worried about attendance at the Olympics in Brazil this summer that they're willing to endanger American lives by not providing better warnings? At the least, pregnant women should be told to avoid non-essential travel to Brazil and Colombia. Anything less is putting political correctness ahead of the well-being of American women. Today, I hope we can gather additional scientific information on Zika and the mosquito that spreads it. I also look forward to hearing about research on the best methods for controlling the spread of the Zika virus. That concludes my opening statement, and the gentlewoman from Texas, Ms. Johnson, the ranking member, is recognized for her opening statement.
Thank you very much, Mr. Chairman, for holding this hearing. This morning, we are talking about the Zika virus, something that has been in the news a lot lately, and that we have been debating on the House floor. The most common way that someone becomes infected with Zika virus is by a mosquito bite. The symptoms of the virus may include fever, rash, joint pain, and red eyes. These symptoms are typically mild, and many people who get infected with Zika virus have no symptoms or do not seek medical treatment. Although the Zika virus was first identified in the late 1940s, and there were no major outbreaks of the Zika virus until 2007. Since then, the Zika virus has been reported in almost 70 countries and territories, including the major Zika outbreak that is happening in Brazil, where more than 1 million cases have been reported. In the United States, local transmission of the Zika virus has been reported in several U.S. territories. In particular, Puerto Rico is suffering from a large Zika outbreak in which more than 800 cases have been reported to date. In the continental United States, the only Zika cases that have been reported involve people who have traveled to places that have a Zika outbreak. But the number of cases is already more than 500 and there's significant concern that the continental United States will start seeing locally transmitted cases of Zika virus once we get further into the mosquito season. Even though my home state of Texas has had 35 travel-related cases of the Zika virus so far, I've been hearing from a lot of concerned constituents about the Zika virus and the potential for a Zika outbreak in Texas this summer. I would imagine that many of my colleagues on this committee have been hearing from concerned constituents as well. I'm pleased that we will have more information to pass on to them after this hearing. What is especially troubling about the Zika virus is that it has been associated with several significant health conditions, including birth defects in infants and neurological conditions in adults. Most notably, the Zika virus has been linked to the microcephaly, a birth defect where the brain does not develop during the pregnancy or after birth. This condition results in significant impairments for the baby and can result in death. Brazil has seen thousands of these cases. In the United States and territories, hundreds of pregnant women who have been infected with the Zika virus are currently being monitored. Unfortunately, last week, the first case of the microcephaly was reported in Puerto Rico. Additionally, the Zika virus has been associated with Guillain-Barré syndrome, a disorder where the body immune system attacks part of the nervous system. People who've had this syndrome usually recover, but the syndrome can result in permanent paralysis. Understandably, people are very concerned about this virus and want more information. That is why I'm happy we are holding this hearing to learn more about the science behind the disease and to learn more about the key research questions. For example, how is the virus transmitted from mother to fetus? And how is the risk of the fetus related to the timing of the mother's infection? Does Zika cause Guillain-Barré syndrome? And if so, if there's an intervention that can prevent this terrible condition. We also need to know where the vector mosquitoes live, the incubation period of the virus in the mosquito, and whether the infected female can transmit the virus to her eggs. Finally, it's perplexing that the Zika virus was first identified in the late 40s, but we are only seeing major Zika outbreaks now. What has changed in the last 70 years to make conditions more suitable for the virus to be transmitted? I imagine that the rapid increase in human travel has played a large role, but I do wonder how much other factors like insecticide resistance and climate change may be playing in the spread of this disease. All of these research questions need to be answered as we plan effective mosquito control programs and prepare to invest our resources. But traditional mosquito control programs will not be enough. I'm looking forward to the hearing from our expert witnesses on how emerging technologies that use advanced genetics and biologic technologies could help control the Zika virus. It is also important to note that to support the necessary research, surveillance, mosquito control, and drug and vaccine development programs that will be needed to control this disease, we must ensure that adequate funding is provided. I'm hopeful that Congress can come together to provide that funding. Time is of essence. And I'm thankful that the experts are here. And just before I yield back, Mr. Chairman, excuse me, I want to take a moment to recognize one of my staff, Kim Montgomery, who is sitting beside me now. She's leaving soon to move to Vienna, Austria. She has been an integral part of the staff, and her passion and expertise will be greatly missed. I want to thank you for your service and wish you luck in your new exciting chapter in Vienna. Thank you, and I yield back, Mr. Chairman.
Thank you, Ms. Johnson. Let me introduce our witnesses, and our first witness today is Dr. Casey Ernst, Associate Professor, Department of Epidemiology and Biostatistics at the University of Arizona. Dr. Ernst joined the University of Arizona staff in 2008. Her area of specialization is the intersection of environment, humans, and mosquito vectors of disease. As an epidemiologist, her role is to work with a highly interdisciplinary team of scientists to integrate information from climology, entomology, medical anthropology, and ecology across multiple institutions to develop an understanding of the emergence of infectious diseases. Dr. Ernst received her master's in public health and doctorate in epidemiology from the University of Michigan. We want to thank particularly Dr. Ernst today for being here, because in so doing she is missing her daughter Savannah's graduation from the 5th grade and on to middle school. So tell your daughter that we appreciate your good help in trying to help the federal government, and hope she'll excuse your missing that graduation. Our second witness today is Dr. Daniel Nisi, Associate Director, Genomic Center for Infectious Disease at the Broad Institute of MIT and Harvard. In this role, Dr. Nisi leads a research group studying the DNA of mosquito vectors and the pathogens they transmit. He has developed pioneering resources and computational methodologies for genetic studies of mosquitoes and mosquito-borne pathogens. Dr. Nisi received his BA from Loyola University in Chicago and his PhD from Harvard University. Our next witness, Dr. Stephen Presley, will be introduced by the gentleman from Texas, Mr. Nogelbauer.
Well, thank you, Mr. Chairman. Our third witness today is Dr. Stephen Presley. He's a professor, Department of Environmental Toxicology at Texas Tech University. Dr. Presley's professional career has focused upon vector-borne infectious diseases with an emphasis on biological threat assessment and countermeasures to protect the population from infectious diseases. He's published more than 90 scientific and technical papers, co-edited 2 textbooks, authored and co-authored 11 book chapters in the fields of biological and chemical countermeasures and diseases of wildlife. He served in the United States Navy as a medical services Corps, and he was an officer for more than 12 years before joining the faculty at Texas Tech University in 2002. He earned his Bachelor of Science in Animal Science from Texas Tech University and a Master of Science and Doctorate of Philosophy in Etymology from Oklahoma State. It's good to have fellow Red Raider, Dr. Stephen Presley, with me today.
Thank you, Mr. Nogelbauer. Our last witness today is Mr. Hayden Perry, Chief Executive Officer of Oxitec, a UK-based company pioneering the use of genetic engineering to control insects that spread disease and damage crops. During his 15-year career at Syngenta, he held various positions, including General Manager of Zika Plant Sciences. Mr. Perry also has served as the European Director and Global Head of R&D for Advanta, one of the world's largest seed companies. More recently, he was CEO of MNL Pharmaceuticals, a company that was focused on pioneering a novel approach to immunology. Mr. Perry received a Bachelor of Arts in International History and Politics from the University of Leeds. We welcome you all, and Dr. Ernst, we begin with you.
Thank you. Thank you, Chairman Smith and Ranking Member Johnson. So today I want to give you a little bit of background on the Aedes aegypti mosquito. This is the mosquito depicted here. It is the primary vector of Zika virus. It's highly adaptable and highly invasive. It's originally from sub-Saharan Africa, but has been widely dispersed across the globe through the mobilization of people and goods. Aedes aegypti is tightly linked to humans and their activities. It prefers to feed on human blood and lays its eggs in man-made containers in and around homes. One of the reasons why it's such a difficult mosquito to control is because it can exploit even as much as 1 inch of water to go through its entire immature life cycle. It's a day biter, which means that it primarily feeds in the morning and in the afternoon, which means things like bed nets are not as effective as protecting against the diseases that it's transmitting. Not only does this mosquito transmit Zika virus, but it is also a primary transmitter of yellow fever, dengue, chikungunya, and one that you probably haven't heard of yet, which is called Mayaro virus. Next slide, please. This is the CDC's recently updated maps of the estimated distribution of Aedes aegypti and Aedes albopictus in the United States. Aedes albopictus is a relative of Aedes aegypti, which is also capable of transmitting Zika virus, but it can withstand cooler temperatures, and it is a more generalist feeder. That means that it doesn't feed as preferentially on humans as Aedes aegypti. Its role in the current outbreak is not well understood at this time. The ranges here are only estimates because many of the jurisdictions in these areas do not have active surveillance for Aedes species. Next slide, please. The predominant transmission cycle for Zika virus occurs when a female Aedes aegypti mosquito bites an infected human. The virus goes through a development stage in the mosquito of a currently unknown time period, probably about a week is what we estimate at this time, and that is dependent upon the temperature. It would be faster if you have hotter temperatures. This is called the extrinsic incubation period. After that time period, the mosquito is capable of transmitting the virus to the next person that it bites, and then remains capable of transmitting for the duration of her life, about 2 to 4 weeks. It's not yet known if the Zika virus can be transmitted through vertical transmission. This means that an infected female mosquito could transmit the virus to her offspring. If it proves to be true, the female Aedes aegypti could emerge infectious from their immature aquatic state and be able to infect people the first time that they bite, shortening the transmission cycle. It would also mean that from season to season, eggs that had overwintered could potentially harbor the virus, and it would not need to be reintroduced the next season. Also depicted in this slide are other transmission modes, which would be sexual transmission from a male partner to a female partner, and then, of course, the transmission from the mother to the fetus. Next slide, please. In order to create forecasts of when and where transmission of Zika virus may occur, there are many components of the system that would need to be understood. We can use weather forecasts to drive models of the mosquito because their biology is largely determined by weather and climate. As you can see in this rather complex depiction here, at each stage of the mosquito life cycle, you have influences of both temperature as well as precipitation and man-made filling of the habitat. Not each of these links is well defined, so we use the best knowledge that we have to estimate the relationships that you see here. The mosquito component of this model is much better understood at this point because Aedes aegypti have been studied in the context of dengue and yellow fever for decades. The top row, which depicts the human portion of the transmission cycle, people who are susceptible, exposed, then infectious, and then recover from the disease, is the least understood part of this cycle. It's also important to note that while this is a theoretical model, there are other things that can mitigate this relationship. Things like vector control, socioeconomic factors, as well as other human activities that could alter what would be depicted coming out of a model. Next slide, please. In the work that we recently published, we initiated the process of exploring some of these complex factors in 50 cities across the United States. What you see in this map is the relative potential abundance of Aedes aegypti as projected from a model using average seasonal climate data. On the top circle, you have the average potential abundance for January, and on the bottom, the potential abundance for July. Some of the areas that have the highest projected abundance coincide with areas where there's been local outbreaks of dengue and chikungunya viruses already. The size of the circle indicates the average number of monthly arrivals from the countries where Zika virus was circulating in February. The tan shading demonstrates the distribution of where actual observations of Aedes aegypti have been made across this area. The work we published is only a first step towards actual forecasting. We need better surveillance, improved knowledge of the virus-mosquito-human interactions, and sustained support for the infrastructure and dissemination of forecasts. Thank you.
Thank you, Dr. Ernst and Dr. Nisi.
Thank you. So, Mr. Chairman, Ranking Member Johnson, and members of the committee, thank you for holding this hearing and for the opportunity to present today. I also want to acknowledge one of the committee members, Representative Clark, an important supporter of the strong research community we have in Cambridge, Mass. My research group at the Broad Institute in Cambridge uses DNA sequencing to understand and control infectious diseases, and I'm here today to speak specifically about how DNA sequencing can help us to understand and inform the control of Zika transmission. We lack fundamental understanding of how the Zika virus moves from person to person via Aedes mosquitoes, information that is crucial for an effective response to this epidemic. Mosquitoes are not flying hypodermic needles that passively transmit disease. The successful transmission of mosquito-borne disease depends on complex biological and ecological interactions between mosquitoes, their human hosts, and the pathogens that they transmit. Insight into the biology of how Aedes mosquitoes are able to spread disease can be gained through sequencing and mapping the Aedes genome, or the entirety of its DNA. We know that because we have demonstrated this with Anopheles mosquitoes, which transmit malaria. So I led a project at the Broad Institute recently, funded by the NIH and involving more than 130 collaborators from 19 countries around the world, to build genome assemblies or maps of all of the DNA of 16 different kinds of Anopheles mosquitoes, some of them very proficient at transmitting malaria and some of them deficient. We learned a lot of things from this project, but if we can advance the slide, I will show just one small result. This is a figure that we built by comparing the genome maps of these different mosquitoes and identifying those groups of genes that were changing most quickly or most slowly between these different mosquitoes. As you can see, the most rapidly changing genes at the top of this list in red are those genes that we know to influence how mosquitoes taste and smell. And we know that there are important genes in this class that determine whether mosquitoes prefer to bite people or animals. And a strong preference for biting humans is a very important determinant of disease transmission. So close behind those smell and taste genes are genes that provide mosquitoes with an immune system. And we know that variation in these genes can determine whether or not mosquitoes are able to transmit pathogens or control infections and not transmit them to the next person they bite. So no comparable set of genomic resources yet exists for Aedes mosquitoes, despite the fact that they transmit Zika, as well as the 3 other major viral diseases that Dr. Ernst mentioned. Next slide, please. Here's an image showing the current state of a recent Aedes aegypti mosquito genome map. The map is in approximately 3,700 pieces, despite the fact that this mosquito has only 3 chromosomes. So the target is 3. The original map for Aedes mosquitoes, published in 2007, was in about 10 times as many pieces, so we're making progress. So as mosquito season begins in the Gulf states, and with Zika on the doorstep of the United States, we lack foundational resources to pursue DNA-based studies of the biology and transmission of Zika. This resource gap is critical. Infectious disease epidemiology has been transformed by DNA during the last 10 years into a rich digital information science, allowing biologists and public health agencies to track the spread of outbreaks over time and space and learn about what mosquito and human factors contribute to disease spread. We can now tackle emergent infectious diseases like Zika using efficient and innovative genetic tools to build databases and situational awareness of diseases. And the scientific community stands ready to develop and apply these tools to the Zika epidemic to protect vulnerable populations within our borders and around the world. One example of that readiness is the Aedes Genome Working Group, organized just this last January via Twitter by Dr. Leslie Vosshall at Rockefeller University. This upstart group's mission has been to produce an improved genome map for Aedes mosquitoes using new sequencing technology that is 10,000 times cheaper than the technology available 10 years ago. With no central funding, but a lot of donated time and resources from a range of academic and industry partners, just motivated to improve our capacity to respond to Zika, this group has made rapid progress exploring a spectrum of new technologies for building an improved genome map. So with continued support and follow-up research, this map will yield knowledge that informs disease control measures. Examples of such measures include insecticide resistance detection and surveillance, mosquito population studies, and genetic modification of the mosquito. New tools for which are creating an increasingly direct connection between basic DNA research and control measures. The Zika epidemic can become a proving ground for the power of new DNA-based resources and epidemiological tools. Taking advantage of rapidly falling costs, we can demonstrate to the world how new technologies will let us understand, anticipate, and control the spread of an epidemic, and we have an obligation to vulnerable populations to seize this opportunity. So thank you to the Chairman, Ranking Member Johnson, and the committee for your attention, and I'll be happy to answer any questions you may have later in the hearing.
Thank you, Dr. Nisi and Dr. Presley.
Good morning, Mr. Chairman and committee members. Thank you for the opportunity to address this important public health threat. My name is Steve Presley. I'm a medical entomologist and professor of environmental toxicology and the director of the Biological Threat Research Laboratory at Texas Tech University. Zika virus, like chikungunya, dengue, and yellow fever virus, is transmitted to humans through the bite of Aedes aegypti and Aedes albopictus. The biology and behavior of these mosquitoes differs significantly from vectors of West Nile virus and other arboviruses in the United States, and these differences pose unique challenges for controlling them in urban and suburban areas. Zika virus infection typically causes less than severe illness, with only about 20% of infected individuals reporting symptoms. Theoretically, a mosquito taking a blood meal from an asymptomatic but infectious individual could become infected with the virus and transmit it to other people, being the initial link in establishing local transmission in an area where no Zika virus cases have previously been reported. Vectors of Zika virus are mainly daytime biters, and they prefer to be in our houses and offices. They are container breeders, and they rest in shaded areas during the heat of the day, in the house or outdoors under the eaves of the house or in vegetation in the yard. My lab at Texas Tech is a component of the CDC's Laboratory Response Network and a certified human diagnostic testing facility. We are a Texas Department of State Health Services testing lab for the Zika virus and other high-consequence infectious diseases. Testing for Zika virus is limited to designated laboratories using a protocol allowed under the Food and Drug Administration's emergency use authorization. The real-time RT-PCR assay that we use detects and differentiates RNA extracted from dengue, chikungunya, and Zika viruses in various bodily fluid samples. The Texas Department of State Health Services is coordinating an entomology consultation group to develop strategies and identify resources relative to controlling the Zika virus vectors. The consultation group is composed of entomologists and public health professionals engaged in mosquito and infectious disease research or public health education. 66% of Texas counties have no records of Aedes aegypti or Aedes albopictus occurrence, and more than 200 Texas counties have not conducted mosquito surveillance during the last 2 years. Scientists of the consultation group are fielding teams to update distribution maps for these vectors in Texas, and there are numerous reports of insecticide resistance in Aedes aegypti and Aedes albopictus populations globally where they occur. The consultation group is identifying resources to determine insecticide resistance in these vectors throughout Texas. The consultation group is drafting Zika prevention information to make available to the public through various online and social networking technologies, providing do-it-yourself information to homeowners on how to eliminate mosquitoes on their property using off-the-shelf commercial pesticide products and application equipment. The American Mosquito Control Association is developing guidelines for mosquito control by public health agencies and commercial entities, emphasizing approaches applicable to the biology and behavior of Aedes aegypti and Aedes albopictus. All of the Zika virus cases in the continental United States have been attributed to travel-associated exposures, with no local transmission confirmed. Conversely, there have been about 800 locally acquired infections reported in American Samoa, Puerto Rico, and the U.S. Virgin Islands, and only 3 travel-associated cases in those areas. This significant difference between locally acquired and travel-associated infections is due to seasonal and climatic differences in most regions. Currently, in most regions of the United States, these vectors' seasonal activity is just now beginning. All efforts must be made to stay ahead of peak mosquito activity by immediately implementing comprehensive public health education programs and enhancing vector surveillance and control capacity. I am collaborating with 2 private companies on projects relative to Zika virus. One company provides a comprehensive digital outcome support system for healthcare professionals, and the other has developed a smartphone-based DNA detection platform that is field deployable and does not require laboratory facilities. Results, the device is a real-time PCR thermocycler that attaches to a smartphone. Results are provided within 40 to 45 minutes and can be tagged with various metadata, synced to a web portal for remote access, and we are planning to conduct field experiments in Florida, Puerto Rico, and Central and South America to validate the accuracy of this system. There's a common realization each time an emerging arthropod-borne disease threatens public health, that being regardless of how modern medical and scientific technologies advance, protecting the public health from vector-borne diseases requires both basic and applied understanding of the vector's biology, behavior, and vulnerabilities. Thank you for your time.
Thank you, Dr. Presley and Mr. Perry.
Thank you, Chairman Smith, Ranking Member Johnson, and members of the committee. Thank you for inviting me to testify today. As the Chairman noted, we are facing an unprecedented challenge of mosquito-borne diseases today. Vector control, controlling the mosquito, is actually our first line of defence, and it's an area we've sadly neglected over the past decades. And because of this, we've seen sharp rises in dengue, we've seen the introduction of chikungunya, and now we have the Zika emergency. And I'm sorry to say, but current insecticidal products are just not sufficient to control this mosquito in the urban environment. So at Oxitec, we looked for and developed a new approach, and that was to use the mosquito against itself. So we have produced a genetically engineered Aedes aegypti strain, which we call OX513A, and it carries 2 genes. It carries a self-limiting gene and a colour marker. Now, the way this works is we release males. Males don't bite, they cannot bite, and they go out and they mate with the wild females. The offspring will all inherit a copy of the self-limiting gene, and then they die before becoming adults, thereby reducing the wild population. And effectively, it's a numbers game. If we can put more of our males into the environment, the females cannot tell the difference. The more will mate with our males, the offspring will die and will bring the population crashing down. The offspring also inherit this fluorescent marker you can see in the slide, which you can't see with the naked eye, but you can see under a light and a filter. And this provides a track and trace type capability. So we can see where our mosquitoes go, we can see what the effect is that we're having, and we can moderate the program as we go along in real time. The releases are made from a truck driven by an operational plan which we work out to make sure we cover the whole town, and we have a GPS system that helps us in terms of decision support. The efficacy of this approach has actually been quite remarkable. In all the outdoor trials we have produced, every single one, we have reduced the Aedes aegypti population in the urban environment by over 90% in about 6 months. And that is hugely more effective than can be done with insecticides. Now, safety and respect for the environment are obviously key factors. And again, in stark contrast to insecticides, we are just targeting the one mosquito species that is spreading the disease. So this is a species-specific approach. Just as important, actually, I'd like to draw 2 points to your attention. Our insects do not persist or stay in the environment. The males we release will die, the offspring will die. It's a matter of days, they will disappear. And also the marker system means actually that the whole process, the whole control program, is precision and metric driven. I'd also like to stress that in contrast to many other approaches that are being mooted at the moment in response to Zika, we actually started this in 2002. Our product was developed in 2002, justified at that time by dengue, and it's all the more necessary now. And then we started doing outdoor evaluation in open field trials since 2009. So we have a very long body of evidence and data to back up both the efficacy and the environmental side. We've now received national biosafety approval in Brazil, and indeed the World Health Organization has specifically recommended this product for operational use as part of their emergency response to Zika. So these are ready to use now. In the US, the regulatory process is actually still ongoing. We opened a file with the FDA as far back as 2011 in order to conduct a small field trial, which is actually a very necessary part of the regulatory requirements. The FDA recently published a finding of no significant impact, but while we're waiting for the final review and the final decision, we're not able to move forward. But in that same time frame, 2011 to today, in Brazil, we have filed similar applications, we've carried out several field trials, we've formed a company, we've built a factory, we've got national biosafety release, and we're now in operational use. Members of the committee, I don't think time is on our side with Zika, and I think the utmost urgency is required in every area. I've come from Puerto Rico, and we could have a catastrophe on our hands if we are not careful. At any one time, there are 28,000 women pregnant, and that is a very salutary thought when you think the summer is coming up and we have active disease transmission. So in view of that urgent health need, I would urge your support, actually, to give the FDA all your support and encouragement so they can expedite approval of our application. And I would also urge consideration of an emergency use authorization, actually, which is used sometimes to bring forward new medication or diagnostics. But I think the fact that we have such an urgent and pressing need means the FDA needs more tools at its disposal in order to help protect Americans. We want to make this technology available in the coming months rather than the coming years. Thank you very much.
Thank you, Mr. Perry. Let me direct my first question to Dr. Presley, Dr. Ernst, and Dr. Nisi. And let me say initially, though, that we have two countries, Brazil and Colombia, where there are thousands of cases of individuals with the symptoms from Zika virus. We know that for every one who has a symptom, there are 20 others who are infected. We also know that over 99% of those in the United States who have become infected and have the symptoms contracted the Zika virus because of travel outside the country. So my question is this: don't you feel that the federal government should issue a travel advisory that says that at the least pregnant women and perhaps others should avoid all non-essential travel to those two countries and perhaps others? Dr. Presley.
Yes, sir, Mr. Chairman. I believe that the potential for people to travel to countries where the Zika virus transmission is actually occurring and then bring back an infection, right. They may be asymptomatic, but I believe that stricter travel restrictions or advisories should be implemented. Okay. When you say that we should suggest that those traveling from the United States to other countries avoid all non-essential, travel, are you talking about just women or a wider I'm talking about any non-essential travel from the standpoint of the few cases of sexual transmission were from male partners that came back, and I know at least one of the cases was a woman. A partner had become infected. So the government should issue a warning that says avoid all non-essential travel to Brazil, Colombia, anywhere else? I believe so, sir. Okay. Thank you. And Dr. Ernst.
I would say that definitely for pregnant women that it would be advisable to have them restrict non-essential travel, but also to make sure that they understand if they are going to an area and they do choose to go to an area, that they look at the geography of the risk. So it is possible that in some of these countries where Zika virus is being transmitted, that there are high altitude locations and other locations within those countries that do not have Zika transmission or even the vector that is present. So really trying to understand more specifically where they're going and working with their doctor to get advisement on whether or not it is important for them to go.
I think I saw a map, at least of Brazil, perhaps of Colombia, that showed over half the country was actually below the 6,000-foot altitude. So it seems to me, but you do agree, at least for pregnant women, that there should be an advisory to avoid all non-essential travel.
Correct.
Okay, thank you. And Dr. Nisi.
Sure. I think this question probably falls closer to the expertise of my co-panelists, but I think it would be reasonable to advise caution, especially for pregnant women who might consider traveling to a Zika endemic area. Okay. But to echo what Dr. Ernst mentioned, beyond geographic considerations and altitude, there are also seasonal dynamics that surely contribute to Zika transmission. So it's
what would be the most dangerous time of the year to travel?
Oh, I think the hot, wet time of year.
Summer?
Yes, okay. But I think at this stage it's very difficult to quantify exactly what that risk represents.
Okay, thank you. Second question to Dr. Presley and Mr. Perry, and that is, what are the best preventative measures that we in the United States could take to try to avoid contracting the virus?
The preventive approaches, because the biology and behavior of these vectors is completely different than the mosquito vectors of West Nile virus or other arboviruses, the immediate concern or the immediate action is, I believe, prevention from the standpoint of educating the public. These mosquitoes are called backyard mosquitoes for a reason, because they like to live around people and in their yards. So the pesticide application technologies and approaches that are used now are pretty much, at least throughout most of the country, is driving down the street in urban areas, dispersing a fog or a spray or an ultra-low volume insecticide that doesn't reach into the backyard. And so the control strategies with pesticides is going to take much more applied when you target an area where active transmission could be occurring. Go in and kill the mosquitoes in the yard, and that may be a homeowner-driven.
Just almost on an individual basis, then.
Yes, sir,
okay.
And it become the individual homeowner until we have a technology like genetic modification, like the sterile screwworm release. Okay,
thank you, Dr. Percy. Mr. Perry, my time has expired, so could you briefly respond?
Yes, I think we can actually target the mosquito now and drive it down to a level where it can't transmit disease. I firmly believe that. I think it's an integrated approach. I wouldn't just rely on our technology, but I think our technology provides the missing piece of the jigsaw. But really we have now, with our technology added into what we have already, a way of controlling this mosquito in an urban environment, and we should focus on the high-risk areas as a priority.
Okay. Thank you, Mr. Perry. The gentlewoman from Oregon, Ms. Bonamici, is recognized for her questions.
Thank you very much, Mr. Chairman, and thank you for convening this expert panel of witnesses. It's been very enlightening. I have about 5 hours of questions, but I only have 5 minutes. So I'm going to start. Dr. Ernst, you said in your written testimony that we don't know the complete distribution of the AG aegypti in the United States because surveillance for the mosquito is not consistent across jurisdictions, and many don't have the resources to carry out the surveillance needed. And Dr. Nisi, you talked about a different kind of mapping, the genomic mapping. So can you each address how close are we? Are we halfway there? Are we 25% there? And maybe just a mention of the where we are in terms of resources as well. And then I want to have time for another question or two.
Sure.
Dr.
So
Ernst.
I will try and answer briefly. So I would say that it's very spotty where we have the surveillance data that is actually accurate and well collected. Some places do a great job and have a significant amount of resources. The majority of places do not. There are West Nile virus vector surveillance that is ongoing, but the distribution, the way the ecological niche is for the Aedes aegypti mosquito, it's different. So you have to have different traps and different location of those traps. I would say at least 80 to 90% of the actual jurisdictions probably do not have active Aedes aegypti surveillance at this point.
Thank you. And Dr. Nisi, on the mapping, how close are we? Or how far are we, depending on how you look at it?
I think we're close. I think we're making good progress towards having a better genetic map, but I think having a better genetic map for the mosquito is a proximal goal, and the goal of applying that map most fruitfully to studies, for example, of insecticide resistance and identifying those markers that are most useful in the field for maintaining the efficacy of the control measures we do have, that will take further application of the genome map and testing of more samples.
Thank you. And many of you mentioned the urgency because of the summer weather that's approaching. It's almost June. Does anyone disagree that it's becoming more urgent as the weather is getting warmer across the country and the world? Anybody disagree with that? So my next question is really for each of you. I was really surprised to learn that the Aedes aegypti mosquito can breed in a bottle cap of water. I don't think that this is what people think about when they're warned against keeping standing water around. And I know, Dr. Presley, you address this need for public health education, but what efforts are underway to educate people? And is there social science research about the most effective ways to communicate these kinds of messages? Are there, do we need more sort of behavioral research on how to send the messages with urgency and so that they'll be understood and acted upon? Dr. Presley, you want to start, and then I'd like to hear from the others as well.
Yes, ma'am. There's efforts underway in Texas, I know for sure, to put out using social media and online resources. There's programs, there's web pages that CDC has. There's a lot of information out there. I did a Zika virus search last week and found 91 million results. So
I don't mean to interrupt, but maybe that gets to the heart of the question. If somebody sees 91 million results, how do they know where to go to get something that's effective? Is the CDC known as the reliable source, and the others are, everything we read on the internet isn't true? So how do we get that message across in an urgent way?
In Texas, in the local area I'm from, we're sending it out in utility, a little flyer, a pamphlet in utility bills. And they're going to do that. They started that in May, and they're going to follow through the summer with that. That's really the only way that we found to make sure everybody at every level gets it.
Okay, I know a lot of people pay their bills online, so I mean, I just don't know if we're doing what we need to do to reach people to get the message across. I mean, I think people are thinking, well, if I have a pond, I'm in trouble, but otherwise I'm not. So how do we communicate that? Dr. Ernst, do you have thoughts on that?
Sure. So we're doing a couple of things. One is to try and engage people in social media, but also to try and link that social media to whether or not it actually makes any kind of difference, because there is a lot of advertising and there is a lot of information that's out there, but whether or not that actually translates into action on a person's part is unknown. We are also developing a mobile application that people can subscribe to that has an educational component. Right. I think that making sure that there is a cohesive on-the-ground network, so in the area where I'm from, promotoras and community health workers can get that information out to areas where there are fewer resources and actually have one-on-one contact with most vulnerable communities.
Terrific. And Dr. Ernst, quickly, you mentioned that we don't know how different environments and different environmental conditions may change the feeding habits. Is somebody researching that, and what would that research tell us? If we could answer those questions, how would we use that information?
Right. So in Phoenix, where there's a fairly good vector surveillance and control program, they actually noted that the Aedes aegypti mosquito were altering their biting behavior and were biting as early as 3 or 4 in the morning. Now, that's anecdotal. They have not yet published that information. But it seems like this is a very flexible mosquito, and it is able to modify its behavior in order to survive.
Fascinating. Thank you, Mr. Chairman. I yield back.
Thank you, Ms. Motomichi. And the gentleman from Oklahoma, Mr. Lucas, is recognized for his questions.
Thank you, Mr. Chairman. And before I begin my questions, I would note that having known Dr. Presley since his graduate days at Oklahoma State, I have a little sense of co-ownership along with Congressman Nagelbauer down there. That said, Doc, let's turn to you for a couple of questions. Number one, in addressing the issues, and the panel's laid out very well today, the challenges we face, the ultimate goal of what we try to do, but there's the short-term solution, there's the long-term solution. Talk for a moment about, and you discussed this earlier, about the mosquito control perspective for a homeowner, whether it's Lubbock or Stillwater or anywhere where this may be an issue. You've described how the traditional mass fogging systems going down the street won't get everywhere. I know historically in the past we've done aerial spraying in certain cases. Tell me, as I talk to my constituents, the off-the-shelf products you're talking about, what kind of cost are we talking about for a homeowner to protect him or herself in their backyard? It's not particularly expensive, correct?
No,
It's the
no.
efforts involved?
No, sir. You can buy a home thermal fogger, which creates a cloud, a smoke cloud essentially, in your backyard that will penetrate vegetation, and you can buy the pesticides. You can buy the thermal fogger for a couple of $100, a very good one. And then the pesticides maybe $20 or $30 a month to keep mosquitoes down in your yard. The issue with these mosquitoes, Albopictus and Aegypti, is they stay pretty close to where they are hatched, about 200 yards, 300 yards in an area. The foggers that drive up and down the streets are really for mosquitoes like Culex tarsalis that vectors West Nile virus, and that with the restrictions and how we've refined pesticide application, you're relying on a small droplet of that insecticide impinging on the mosquito while in flight. That's effective for crepuscular biters or mosquitoes that bite at night or between sunset, dusk and dawn, because with Aedes aegypti and Aedes albopictus, they're daytime biters primarily. So spraying with a fogger depends on before the thermal inversion occurs. When the sun comes up and the ground begins to heat, the currents are upwards. So these fogs, these clouds that you spray out of a ULV machine driving up and down the street will rise and dissipate. You're trying to get, with these daytime biters, treating early in the morning with a fogger or a thermal fogger or a spray, just a wand spray, treating under the eaves of houses, around doors and windows and vegetation. It's a completely different strategy for vector control. It comes into personal or private property issues, all of that. The homeowner is going to be responsible for dumping that bottle cap full of water, or that toy dump truck in the backyard, or anything that can collect water is a good breeding site for these mosquitoes.
But our fellow citizens have the ability to protect themselves if they'll take action based on the right information.
Yes, sir, and there are numerous over-the-counter, off-the-shelf products that can be bought at any big box garden store or home improvement center.
Now let's go to the next step. Mr. Parr is discussing an effort at a long-term solution. You mentioned the screwworm program, so successful since the 1950s that most people don't even know that it exists. Would you expand on the problem we faced in the Southwest, in the South for that matter, what the solution was and how it has continued to be effective since then?
Oh, the sterile male release of screwworm flies. The primary screwworm, they're a blowfly. They would oviposit or larvaposit their maggots into a wound, and that particular screw fly would eat living flesh. Other blowflies will just...
And any cattleman 65 years or older in Texas or Oklahoma will tell you it was a horrible thing.
And not only cattle, wildlife, white-tailed deer, everything was affected. They came up with a sterile male screw fly release program where they irradiated the males and released them. This particular species only bred once, the females, so it stopped the progression, and it essentially stopped.
For 50 years now.
There still are cases, but it's very controllable and managed on the Texas border now.
So we have a track record of using these type technologies to make a real difference, systematic and in the long haul.
Yes, sir.
Thank you, Doctor. Yield back, Mr. Chairman.
Thank you, Mr. Lucas. And the gentleman from California, Mr. Bear, is recognized for questions.
Thank you, Mr. Chairman. I'm going to put on my doctor cap here and public health perspective. So with regards to travel advisory, for those listening at home, just go to CDC Zika Travel Advisory, and you'll get a lot of information and country-specific information. Going back as far as February, the advice has been, if you're pregnant, do not travel to endemic areas. If you're of childbearing age, the advice is not to travel to endemic areas, but if you are going to travel, to take precautions, birth control, et cetera. We've also been working with global health agencies to increase access to birth control in endemic areas until we get ahead of Zika virus and we fully understand what's going on. If you have to travel to these areas, obviously take mosquito precautions, repellents, et cetera. Avoid going out, at dusk or the times when the mosquito's biting. In addition, back in February, we had a hearing. We had Dr. Frieden and Dr. Fauci come over from the National Institutes of Allergies and Infectious Disease. Dr. Fauci at that time was quite optimistic by the end of the year that we'd have a vaccine for clinical trials. I will be honest, I was skeptical that we'd be able to develop something as quickly as that. A few weeks ago, I had a chance to go visit Dr. Fauci and his team at the National Institutes of Health. They will be ready for clinical trials, phase 1 trials, starting in September. And a very important aspect of our getting ahead of Zika is that vaccine development and the science behind vaccine development. So, September's still after mosquito season, but we are moving fairly rapidly to get ahead of this and address it. The bigger component here, though, and each of the witnesses touched on this, is 2 years ago we were talking about Ebola, we're talking about Zika this summer, and the world is a much smaller place right now. And vectors, infectious disease, viruses move much more quickly. It's not as though we haven't known about Zika for years. We've known about this since the 40s. And it's not that the virus has mutated. The virus has moved. It's now in a densely populated area. And we are starting to see this pop up. And what we have to do as a body and as, is provide adequate resources to the CDC, to the NIH to do the research, but to understand that we are going to be battling these infectious diseases much more frequently, and we've got to support the science. Dr. Ernst, I'd probably toss it over to you with regards to both Zika, certainly as we get a vaccine developed, disseminating that vaccine and making sure it's effective, but then also just more broadly as an epidemiologist and someone who studies this, what we ought to be doing right now to prevent the next infectious disease and how we ought to be thinking about some of this.
So you're talking more broadly, any infectious disease?
More broadly, yes.
More broadly. I mean, I think some of the things that we can do is really understand where a lot of these infections are originating from. A lot of these are diseases of poverty. They're diseases where you have countries and places that have no infrastructure to be able to respond and control the epidemic in their own boundaries. And when you have something that is going on in another place and you have, as you said, a very globalized world, it doesn't take a lot for those viruses or bacteria to come over here. So I think shoring up resources in other places to ensure that there's fewer pandemics in those areas as well is a critical step. And then also investing in research and understanding where those threats might be more likely to come from is also very important.
It's wonderful. And again, I think for the public and for my colleagues in Congress, it is very important that we don't just fight the disease here, that we actually go to where these infections and these viruses are endemic and originating. And there's a reason why we fund global health. There's a reason why we fund the CDC to go do research and go to these endemic areas. And as we look at these fundings and we look at our budgets, it's really important for us because if we don't get ahead of this in those endemic places, where the viruses, they will certainly pop up here. And we've seen it time and time again with SARS, with various infectious diseases. And again, in a smaller world, we will see this more frequently. I'm running out of time. As Ms. Bonamici said, I could spend 5 hours asking questions. But we also have to then adequately fund the science as well. So we come up with those therapies to both control the vectors, but then also to get ahead of this. And with that, Mr. Chairman, I'll yield back.
Thank you, Mr. Bear. And the gentleman from Texas, Mr. Nagelbauer, is recognized.
Thank you, Mr. Chairman. The United States has been very lucky that many of the potential global health pandemics have not become epidemic in the U.S. However, it's a little puzzling to me because, some of these things like Ebola, Zika, have been around for a very long time, but what happens is something kind of elevates their attention, And then there's a call then for the federal government to come and spend billions of dollars to be in a reactive mode. So I guess my first question to the panel this morning is, rather than being in a crisis mode on some of these things, what would be a better plan? And because I think what I always find is when we have crisis, we don't spend money as efficiently as we do when we spend money when we're planning for it. So kind of go down the panel and in a perfect world, how can we get on the front of some of these rather than being in a reactive mode?
So I would concur with that. I absolutely agree that the reactive mode that we seem to be in terms of pandemic response and planning is not as efficient. I think we have made some strides towards trying to be more ahead of the curve. We had a lot of preparedness funding that has come through the health departments to try and set up some of these response plans in place, et cetera. But I think we can do more. I think that we need more investment in trying to understand where and when and forecast some of these risks and be able to quell them before they can even actually start taking off. And that means having really better surveillance systems globally as well as even improving our surveillance systems here domestically.
Right. So, I second the the importance of surveillance, and I can speak to work being done by one of my colleagues at the Broad Institute, Pardis Sabeti, who was well positioned to apply DNA-based studies of the Ebola outbreak because she was establishing a research base in West Africa and starting to characterize fevers of unknown origin. There are many agents of infectious disease that result in symptoms and that are undiagnosable because we don't know what the agent is. And there are many of these, particularly in tropical parts of the world, any one of which could, in principle, emerge to cause an epidemic. So I would advocate the importance of surveillance, the importance of catching new epidemics early, and the utility of DNA-based studies for getting an early characterization of perhaps the agent behind emerging outbreaks.
I agree completely with what's been said. I'm much more applied in how I study vectors. It's more not at the genetic level, it's more at transmission dynamics, how they behave, the ecology of the vectors. That is where I believe funding also needs to be focused. You're right, this everybody panic and throw money at it doesn't fix it. And we have, I think I said it before, Zika virus is just the latest of an emerging arthropod-borne threat, public health threat in the United States. We had chikungunya last year, Rift Valley fever before that, and we'll have more because there are viruses out there that we know or suspect are vector-borne, but they're not in areas where we're really concerned right now. West Nile virus had never occurred in the real world until 1999. Zika virus wasn't in South America until last year, 2015. So viruses are moving around, and that's part of whether it's climate change or global travel, speed of travel, they're moving around. My basic belief is that we need to start at the applied level and train pest control operators. We saw a knee-jerk response to Ebola where nurses and clinicians did not have the proper personal protective equipment, the most basic of supplies they need. I can draw a similar situation with vector control operators in mosquito control districts throughout the country right now. They don't have those basic supplies they need, knowledge-wise and resource-wise, to attack this issue.
I think you have to look at the common element, and the common element is the mosquito. Control the mosquito, you control all the diseases that it has spread, is spreading, and will spread. And when I mean control, I mean giving yourselves the capacity, the tools, and the procedures to exercise that control. And in some areas, one may wish to eliminate the mosquito. In other areas, one may wish to have the ability to drive it down below a disease transmission threshold, which is not actually the same thing as elimination. But having that capacity, having that ability to do that, to me, is the absolute starting point of this.
Okay. Thank you, Mr. Nagelbauer. And the gentleman from California, Mr. Takano, is recognized for his questions.
Thank you, Mr. Chairman. Mr. Perry, what you propose makes rational sense. Statistically drive down the numbers of the mosquito control, the vector. Mr. Lucas mentioned the sterilization of the male insect. Your process, though, is not sterilization. It's about a genetically modified mosquito. And there are some concerns of some environmental groups about the safety of this. And there's some rumors that this genetically modified mosquito actually transmits the virus. Can you just quickly clear that up, because I have a lot of other questions I want to ask.
Yes. I mean, our mosquito is modified in two ways. It can't reproduce, and it carries a colour. It's no more or less capable of doing anything else than a normal mosquito, so there is no difference. In terms of the environment, actually, I think our mosquito is far safer and softer on the environment than any other intervention. It's not a toxin. You're just taking out one insect. You're not affecting the rest of all the food chains. I think it is a shame, in many ways, that there has been a concern over the words genetic modification in society. That's been there for several years now. And really, we need to be able to distinguish products by what they are, what they do, and what their risk profile is, rather than assign a tag or a label. But ultimately, with our product, it's like any other. We provide the evidence, but the actual arbiter, at the end of the day, you go through the regulatory process, the evidence is weighed up, and you have an outcome that's based on science and facts.
So with respect to Zika in this particular case, it seems controlling the vector is really important to keep what transmits the virus from doing that. And so reducing the numbers of mosquitoes is really important, but also coming up with better technologies on repellents. In my own district, the University of California, Riverside, which is known as a great center of entomological research, I see a lot of nodding heads. Dr. Nefsi, are you familiar with olfactory laboratories, which have an attempt to monetize the research that was done in a laboratory by Dr. Ananda Sankar Ray, who has basically focused on trying to deal with the carbon dioxide receptors of the mosquito. You mentioned that some mosquitoes are able to distinguish between human and animals, but what's common is that they're kind of attracted to this carbon dioxide marker. Is that right?
So yes, that's right. I think there are a number of cues that lead mosquitoes to their host for a blood meal. Carbon dioxide is one such cue. What is amazing is that many mosquitoes, particularly those ones that are most culpable in transmitting human diseases, are very human-specific. So they're responding to carbon dioxide, but they're also responding to other unique signatures of human odor. I think developing repellents, developing strategies that capitalize on our rapidly growing knowledge about the molecular biology and the neurobiological basis of how mosquitoes perceive these cues is a fantastic research direction, and I think you've cited a great example of how it's actually translating into intervention measures or repellents.
And this is a great example of how basic scientific research in a laboratory has these very practical effects and is being furthered by private sector funded efforts to monetize that research. I understand that they've developed a patch, something called the kite patch, which can just be worn on clothing, which somewhat interferes with the mosquito's ability to perceive these, what do you call them, these markers or
the cues.
The cues, the chemical cues. What's exciting about that is it seems has less impact on the environment with the use of these pesticides. So this is a very promising line of research, is what I perceive.
Absolutely. I think we're positioned now, rather than to use sort of a random strategy for finding chemical repellents or other interventions. We can really use our knowledge to pursue these strategies with a rational basis, with a thorough understanding of the mosquito biology, and these have great potential.
Well, I hope that we can work together on both sides of the aisle to promote a number of strategies to deal with this vector. Mr. Perry, you certainly proposed, I think, a very rational strategy, and Dr. Nefsi, you've confirmed my excitement about what's going on in my own district in this research, and to the extent that we can buttress what's going on out there. And Dr. Ernst, what you say about global poverty and making sure that we take care that there can be no weak link. We can't allow any country to have a dysfunctional public health system, and it comes back on us. I yield back, Mr. Chairman.
Thank you, Mr. Takano. And the gentleman from Louisiana, Mr. Abraham, is recognized.
Thank you, Mr. Chairman. I think we certainly have a great panel here, and with your minds and the minds out there working on this, we can probably cure this issue. Going back to Congressman Neuberger's argument of being reactive instead of proactive, the federal government is unfortunately often reactive, in my opinion, and it's like sending the Hindenburg to rescue the Titanic. We need to be better than that, and we should be better than that at this stage of the game. Mr. Perry, the CDC, I think in February, elevated Zika to level 1 activation, and you can help me, but I think I recall that the only other time is Ebola was active 1, H1N1, and being in Louisiana, I remember post-Katrina was a level 1 activation. So my question to you is, you've got evidently something that works here. You said you could get a 90% reduction in mosquito population in 6 months, which is, to me, a phenomenal feat. If CDC thinks this is so important, and I can assure you it's very important, I've treated microcephaly and microencephalitic children, and it's a horrible, horrible thing for a child to undergo that, and the parents also. What is the FDA telling your company as to why it won't give you an emergency declaration? I mean, you've been after this since 2011, according to your testimony. We know this is bad. We don't want any child to be born with microcephaly or microencephaly. What's the answer FDA is giving you? Say, why can't we do this now?
The answer is that we have received is it's complicated.
Well, come on, it's not that we've got wonderful scientists here, including yourself, that tells us we need to act now. We should have acted a year ago.
I absolutely agree. We're being treated as an investigational animal drug, as the sort of product class, if you like, because the genetic modification is a DNA insertion. Which means that from the FDA perspective, they would have to approve an animal drug in order to provide a public health benefit.
But you've got objective data that says this works now.
Exactly, and I think that's exactly the way we should go. We need to go down an emergency.
Well, we've got to kill these mosquitoes. I mean, I understand, look, I think it's critically important what we've talked about, better repellents like Mr. Takano said, certainly spraying individually, but until we kill the mosquito, we can kill all we want to in our house, but they're going to come back in. Mosquitoes are going to come from the islands, they're going to come up from the south. We've got, again, we've got to quit putting Band-Aids on this, and we've got to go to the root cause. The root cause, like you alluded to, is a mosquito. And somebody asked about pandemics, epidemics worldwide, and again, yellow fever, dengue, malaria, all these are vector-borne, and we've got to kill the source. So, I'm very frustrated, as I am with many government agencies, unfortunately, but with the FDA in this instance, it's out there, we know it's here, and we need to do something, but they're putting up blockages to your companies and other companies like yours.
I think we should encourage them to find the processes to make this happen.
Is the cost of your technology any more costly than spraying, a traditional spraying?
I think in terms of cost efficacy, ours will be streets better, to be honest.
Really? Okay. And to the panel, I've just got a couple of science questions. Is this a intracellular or an extracellular virus? Does it live in the RBC, WC, or is it in the plasma? Where does it live in the human?
I think it replicates in cells, and I think work is underway on elaborating exactly which cell types are most favored by the virus. I think there was this convincing work a few weeks ago with brain tissue organoids to establish its affinity for developing neuronal cells. But I would say that the full preferences for the types of cells infected by the Zika virus are still being elaborated upon.
And Dr. Ernst, you said that certainly the mosquito vectors like the Aedes aegypti and the Alopecia transmit not only Zika, but other diseases such as dengue, yellow fever. When a mosquito that's infected with Zika and those other diseases bites somebody, are both viruses or more than one transmitted at the same time, or does Zika take precedent? Who wins that battle?
That's a good question. I don't think we know that yet. There's certainly some evidence that mosquitoes can be co-infected with dengue and chikungunya, but I'm not sure what the status of the research is right now on the interactions between dengue virus, for example, and Zika virus. I don't think we know that yet.
And one quick question, my time is out. After somebody's infected with Zika, is there antibodies built up? Can we test for antibodies in that person?
Yes, we can test for antibodies for Zika, but it's very difficult to disentangle if the person has had a previous dengue infection. Then when you try and test for antibodies, it becomes equivocal. The results become very difficult to interpret.
Okay. Well, Mr. Chairman, I have 1,000 other questions also, but my time is up, so thank you very much for allowing me to go over.
Thank you, Mr. Abraham. And the gentleman from Colorado, Mr. Perlmutter, is recognized.
Thank you, Mr. Chair. This is a very fascinating hearing, and obviously, my question is going to be a little more parochial. From Colorado, looking at those maps that you presented to us, we're not really in the region that can anticipate Zika, the Zika mosquito, the Aegyptus, or whatever it is. So I'd like to start with you, Professor Presley, because you mentioned West Nile, which is something that we'll get a couple cases of West Nile in Colorado pretty much every year. Can you just, for a layman like myself, kind of explain the difference between the mosquito that delivers West Nile versus the mosquito that delivers the Zika virus? And then, you know, how are we managing West Nile mosquito versus the Zika mosquito?
The West Nile vectors is and I'm not sure what part of Colorado you're talking about.
Denver area.
On the high plains,
yeah.
It's Culex tarsalis.
Okay.
And that mosquito breeds in pools and ponds, standing water, so we can survey for them. Their activity is primarily crepuscular, right after dark and right before sunrise.
Okay.
Contrast that to Aedes aegypti and Aedes albopictus that are vectors of Zika virus. And they're container breeders. It's fresher water, typically in small containers, in the axils of plants.
When you say small container, yeah, tell me what you mean by small containers. I'm trying to think, what do we ship them in boxes or what?
A tuna can is more than an empty tuna can is more than enough water. They will breed in the leaf axils of bromeliad plants, like in the house. You know, a comment was made about just outside. No, these mosquitoes will live in the house. They like to be in the house, hotel rooms, so just treating outside doesn't control them. So these differences in where they breed and where they like to bite and rest are significantly different. You know, they're daytime biters, and it's really right after sunrise, typically right after sunrise, then they rest during the heat of the day, and they bite right before sunset when it cools back down. That's not a rule, but that's generally.
So in controlling them, and Mr. Perry, maybe I'll direct these questions to you for a second and then to the whole panel. So, and I don't know the Latin term, but I'll call them the West Nile mosquito and the Zika mosquito. In your use of more or less a sterile male to defeat the virulent female, or, you know, cause the death of the species, do you guys do any work on the West Nile mosquito?
Not yet. So we've obviously developed Aegypti. We know we can do Anopheles, which is the malaria mosquito. We've actually done proof of concept work there. We're actually developing Aedes albopictus, which was also mentioned earlier. So that is actually coming through the R&D system and as a candidate product. With the Culex mosquitoes, the West Nile ones, we've looked at those. We think they're very tractable, actually. We can do them. The research team is very confident, but we haven't proven that yet.
In Colorado, I think, Professor Presley, we have up at Colorado State University, you know, up in, studying the Zika virus, and and the mosquitoes. I guess my question is, the symptoms of West Nile can make you get really sick. More negligible, obvious symptoms with the Zika virus, but the potential damage to the fetus or to the baby is tremendous.
Yes, sir.
So I guess my question is, similar to Mr. Abraham's, we ought to be allowing every possible defense, whether it's using an insecticide or sterility, and I would, on the insecticide part of this, do we have any breakthroughs? It sounds like Mr. Perry has a good approach on the sterility. Do we have any breakthroughs on the insecticide piece of this against the Zika virus and the Zika mosquito?
We do, Congressman. There are some very effective pesticides, but the whole insecticide resistance issue in these species is critical, and we don't know where resistant populations are. Texas, the state of Texas, we're going to find out in Texas, but there needs to be, I think, a national effort where these mosquitoes are documented to occur to find out what insecticide resistance does exist in the populations so we can knock them down immediately and then rely on long-term strategies to keep the numbers down. And I would just add in that West Nile virus, when it first occurred and during the first few years of its occurrence in the United States, was considered to be West Nile fever predominantly, which was relatively mild symptoms. Now it's predominantly West Nile neuroinvasive disease. The virus has changed over time. We don't know what Zika is going to do because it's only been in the New World in South America for a year.
Okay.
There are a lot of challenges there.
All right. Thank you, Mr. Chairman.
Can I add a point?
Oh, go ahead.
Sorry, I know that's not done, but I'd just like to add a point. The issue of insecticide resistance is a very real one. But when you get to the Aedes aegypti, which is the Zika transmitter, the biggest issue is private property, actually, because this mosquito lives in and around the home.
Okay.
For chemicals to be effective, you've got to have a public health authority coming into your house, your child's bedroom, your kitchen, pretty much every week. And that doesn't work with modern society.
Okay, thank you.
Thank you, Mr. Perlmutter. And the gentleman from Alabama, Mr. Palmer, is recognized for his questions.
Thank you, Mr. Chairman. Dr. Presley, given that the symptoms of Zika virus in adults are associated with a host of other elements, how likely is it that a general practitioner may not have seen a case of Zika and therefore be able to accurately diagnose it?
Excellent question, Congressman. You know, we say that the facts that are out there or the information out there is that only 20% are symptomatic of infections. Flu-like illness or influenza-like illness is so common with talking to my physician friends. How many infections are we saying, it looks like a virus, go home, rest, plenty of fluids, you'll be fine, and it never gets tested. So I think that these estimates of the number of infections in an area are way underestimated. But there's no way that you can nail that down. It's like West Nile or flu, influenza.
Do the physicians have the necessary diagnostic tools? I mean, is there something out there that they could use to make sure they make a more accurate diagnosis?
The current restrictions on human diagnostics, and my lab is one lab that does that, but there's certain criteria, both clinical and epidemiological criteria, like have you traveled or been exposed to somebody that did travel in a Zika infectious area. And then the local public health authority has to authorize that test being performed. Those tests, the diagnostic assays, can only be done at CDC LRN laboratories. And we've done a couple over the past few weeks, but it's a long administrative trail.
Sounds like we need a kit.
Yes, sir.
Something like that. Let me move on. Thank you for your answer. Dr. Ernst, in your testimony, you described the Zika map that you helped develop to try and predict where Zika might spread in the United States. However, you stopped short of calling it a forecast because there are still too many unknowns. How would you improve your model to provide a better forecast for Zika or other vector-borne diseases? And what resources would a team need to provide a better forecast?
So I think there are several key components that would be helpful to improve for model results. One is being able to have better validation. So we were lucky enough to find a couple of data sets to validate our model outputs. But as has been mentioned multiple times, having actual robust surveillance programs that allow us to test our results against the actual data is one step. Another step is having more information on the specifics of the host and vector interactions. So critical are things like the extrinsic incubation period. We don't really know what that is. We don't know its temperature dependency. We do have some work that's going on in one of my collaborators' labs to identify that. And some other issues are really understanding the social side. So there are absolutely different factors like poverty, vector-human interaction that need to be incorporated into models in order to really predict that risk.
I want to move to another question, Mr. Pari. We banned DDT, and by some estimates, there are 50 million people have died from mosquito-borne diseases. And just in regard to the new science that we're trying to apply to this, what are the odds and potential consequences of the offspring of Oxitec, genetically modified mosquitoes developing resistance or tolerance to the self-limiting gene they acquire?
We haven't seen it. We've gone through over 200 generations now of our mosquito. We've seen no resistance developing at all. There are certain biological issues and also the way in which the technology has been done. So we're not expecting to see it, but any scientist will say you'll never say never. I think the issue is, what does our product actually do? It stops the mosquito from breeding. So what happens if it doesn't work? Well, then the mosquito can breed. So it becomes a normal mosquito. So effectively, we would stop releasing.
Well, looking at the situation from another angle, is there any chance that Oxitec technology might eradicate the Aedes aegypti breed of mosquitoes?
Well, our mosquitoes only fly about 200 yards in their lifetime. So actually the effect you get when you're releasing our mosquito is in the area in which you're releasing it. So you actually have to be very deliberate about where you go, how you control it. And against all of the issues you've raised, we have the marker. We have something that no one's ever had, which is the ability to track and trace exactly what we're doing. So we pick up that metric all the time.
Well, I'm out of time. My last question here is one of our colleagues on the committee talked about CO2 as a marker, apparently in the context of climate issues. Is it any of your recommendations that we breathe less? I yield back.
Thank you, Mr. Palmer. The gentleman from New York, Mr. Tonko, is recognized.
Thank you, Mr. Chair, and welcome to all of our experts for very valuable information. Dr. Ernst, in your testimony, you discussed community-based surveillance activities, including USDA's efforts to encourage participation by community members. There's no doubt that we need to invest more resources into surveillance. In particular, I'm encouraged by efforts to use crowdsourcing and citizen science methods within the federal government to advance and accelerate scientific research and literacy. One example of this is USDA's Invasive Mosquito Project, the AMP, which was launched recently as an initiative that pairs high school teachers and students with mosquito control and public health professionals. This partnered citizen science classroom project helps high school teachers meet the next generation science standards and students learn about mosquitoes, public health, and safety. The project is aimed at monitoring invasive container-inhabiting mosquito species across the United States. According to the project's description, by doing this monitoring, we can determine where the invasive mosquito species, as well as the native species, are distributed across the U.S. and define at-risk human and animal populations based on this distribution. With all of that said, can you speak more about why projects like these can help with surveillance efforts as well as providing enhanced public education?
Sure. So I think that these kinds of projects are great for a number of reasons. We've carried off some of these in Arizona specifically as well. We developed something called the Great Arizona Mosquito Hunt, and that engaged high school students to set out the oviposition cups, which is a fairly sensitive measure of detecting whether or not there were mosquitoes present or not. And it allowed them to go through an educational module to teach them about the mosquitoes. So I think these kinds of efforts not only can be broadly disseminated to a large group of people, but it's also targeting young children as well as teachers who can pass that information on to their parents and really help be vigilant about mosquito control within their own household. I think that there is some things that need to be ensured for these kinds of projects, including standardization of the methods to ensure that when you do get a negative, that it is because there is not a mosquito around. That's one of the things that's difficult to determine. We found a lot of negatives in the high schools where we placed traps, in part because, as the gentlemen have said previously, it doesn't fly very far, and school grounds are generally kept pretty clean. So really understanding some strategies to standardize this process to yield the best success are also important.
Thank you. And to you also, Dr. Ernst, in reading testimony for today, I was struck by how we do not have a good idea where the mosquito that transmits the Zika virus lives. You have done research into modeling the geographic areas with higher potential risk for local Zika transmission. Could you please describe that study and how forecasting studies like yours could help decide where to invest our resources?
Sure. So this was sort of a first cut, I think, is what we basically have been stating in trying to understand where the Aedes aegypti might be present in the United States, as well as the seasonality of the vector. And it really is based upon what we know about the dynamics of the mosquito and how it relates to climate and weather. So most of the stages of the mosquito are sensitive to temperature as well as precipitation for the aquatic stages of the mosquito. So we can leverage information on sort of average. What we did was we took average 10 years worth of data to look at if you had the weather that's sort of typical in these cities, when would you see Aedes aegypti abundance? And then we compared that to an area where we know there's fairly good conditions. We compared it to the numbers that we had in Miami. And so the map that we actually depict is relative abundance. So it's relative to sort of the high season in one of the most climatically suitable places. And we look to see how different that might be for the other 49 cities that were mapped and modeled. I'm sorry, I can't remember the second part of your question.
Well, it was just how we could best use that information to decide where to invest resources.
Right. So, some of the other layers that we have incorporated are related to travel introduction. So obviously, without any local transmission at this point, it would have to come from somebody who had traveled outside the country and brought back the virus. And then as well, needing to understand poverty and other factors that might facilitate more vector-human interaction. Some of the things that you can use the data that we presented for are understanding when, theoretically, you're going to have a higher peak of mosquito activity within your jurisdiction if you're on the map, as well as trying to look to see, has your jurisdiction actually had dengue or chikungunya in the past? That's another piece that we mapped into that study.
Thank you very much. I sense, Mr. Chair, that there's a degree of urgency to invest here and invest wisely and deeply into this program. If I might take the liberty, today is an opportunity for foster youth to shadow individual legislators. And if I might introduce right behind Congressmember Clark, we have Stephen Fallon, who is my shadow today as a foster youth. If you'll recognize Stephen, please.
Thank you, Mr. Tonko. And the gentleman from Michigan, Mr. Molinar, is recognized for his questions.
Thank you, Mr. Chairman. Dr. Ernst, in your testimony, you identified some of the knowledge gap areas about the interaction between the Zika virus and the mosquito that spreads it. I was also curious, Dr. Nifsi, the work you're doing to sequence and map the genome, is that going to help fill in some of the gaps in knowledge and kind of where are we in that process, timelines, those kinds of things?
Sure. So I think I can attest to the value that genome sequences can have as a resource for informing precisely the kinds of parameters that make the types of models Dr. Ernst described accurate. In the malaria field, we've been using this kind of data to inform models of malaria transmission and malaria distribution. One example of the way these data can be used is a study that was published last year by investigators at Notre Dame who did genetic profiling of Aedes mosquitoes from the Capitol Hill neighborhood and used the genetic signature of these mosquito samples over the course of several years to determine that this was a year-round population, likely a year-round population, taking refuge in sewers or below ground during the cold winter months, rather than a new population that was being refounded by genetically different sources with every successive warm season. So this is a small example of some of the resolution that DNA-based data can lend to some of the parameters that are useful for understanding the existing and maybe the prospective distribution of mosquitoes.
And I was just reading a little bit about, you're kind of communicating via Twitter and periodically getting together? Are there like kind of work projects in place, and how do you monitor who's doing what? And
yeah, so Twitter is a fantastic scientific resource. I don't know if it's recognized outside of the scientific community, but news travels so quickly. It is unparalleled for organizing people who are like-minded and who want to get behind a common cause. So Twitter, I think, and a lament about the quality of the existing Aedes mosquito genome map was the genesis of this working group I alluded to. Since then, we've had phone conversations, emails, but as yet, no in-person meeting. As I mentioned, this is a kind of a volunteer group, and we've been working catch as catch can to this point.
I know you don't want to commit to a timetable of when, the completion of the assembly of the DNA map, but can you give us a rough ballpark?
I can say that we have parallel efforts underway using several different new technologies that we all hope will yield a better genome map on the order of a month or two. Okay. These are unproven, but we are very optimistic given their performance with sequencing human genomes and assembling maps for human DNA. So I think we're optimistic that we won't have a perfect genome map by this fall, but we will have a map that is significantly improved beyond the existing one.
Okay, thank you very much. I yield back, Mr. Chairman.
Thank you, Mr. Molinar, and the gentlewoman from Massachusetts, Ms. Clark, is recognized.
Thank you, Mr. Chairman, and thanks to all the panelists, especially Dr. Ernst. Thank you for making your decision to skip that milestone. We appreciate the information that you all are bringing to us today. I want to just start with something very specific and sort of move to the more general. The impact of this virus on a fetus is one of the most frightening aspects of a rapidly changing landscape. But do we know now exactly how the virus causes birth defects like microcephaly, and do we have an understanding of how exposure at different times of pregnancy can impact the fetus? Or is this a gap in our research?
I don't think we have a virologist on the panel, but I'm going to step slightly outside my comfort zone just to provide a basic answer and say that the general principles that determine what cells and what hosts a virus can infect often come down to the particular proteins or other attributes that are on the surface of those cells. The virus usually needs to recognize a doorway and be able to interact with it in a way that permits entry. So some of the work that's been done to date has taken advantage of some of these fantastic new resources in the laboratory, such as these brain organoids, very simple, very small, three-dimensional tissue replicas of what a developing brain might look like. And the realization that has led to is that there are particular cells at stages of brain development that are indeed susceptible to Zika infection. I think there's much work to be done to understand, as I mentioned previously, the spectrum of cell types that could be susceptible to Zika infection and the precise proteins and sort of molecular interplay that's responsible for Zika's ability to infect the cell types and the range of organisms that it does infect. On the host side as well as, let me say, the mosquito side. Its ability to proliferate within mosquitoes and be injected into people that are bitten subsequently is another aspect of the molecular biology that I think we need to determine.
Yeah, Dr. Presley.
If I could add, ma'am, there is so little known right
Yeah.
now about how the Zika virus interacts in the body, because we all looked at each other
Right.
and shook our head. You know, something to consider, and it's another area that we're going to have to find out about, is, you know, you may not be able to find it in a person's blood after they've recovered a week or so after being viremic. You might not be able to find it in blood, but they're finding it in semen 60 days after recovery or no more symptoms. The virus is going somewhere, hiding somewhere. There's a lot of viruses that do that.
Yeah.
Whether it's deep organ or whatever. I think there's a lot to learn. I think we can agree on that.
And I think that brings me to a more general point that may be even harder to answer, especially in under 2 minutes. But as we have a discussion about how we react as Congress and in funding, and when we get into pandemic mode, do we not make the most efficient choices? But here we are with this very frightening virus that is only one potential just mosquito-borne virus that has changed very quickly, spread very quickly around the globe and now in the United States. And we have very little understanding. And as I've listened to your answers with the need for diagnostic, looking at resistance, mosquito control, dealing with issues of social behavior, private property, how we balance those, also looking at vaccines, understanding the genome, on and on. How do we approach these? And I have some concerns about the way we have funded our response by taking it from funding from other important diseases. And what is the better way? One of the most terrifying conversations I ever had was at the Broad looking at antibiotic resistance and infections. So how can we do a better job of funding basic science that allows you to do the applied science that can bring us some answers in 22 seconds?
I can speak briefly. I think what's important is to set up an infrastructure for collecting and pooling information. I think there are a lot of investigators collecting diverse forms of information, and I think there are opportunities to really rapidly advance our state of knowledge about Zika and other emerging epidemics on the horizon by organizing that information, creating communication channels, not just exclusively via Twitter, but other media, and making sure that we are performing surveillance, that we're collecting the kind of information we need to, in the short term, limit the advance of these diseases, and in the longer term, understand them so that we can develop longer term control measures.
Great. Thank you. I see I'm out of time. I yield back.
Thank you, Ms. Clark. The gentleman from California, Mr. Swalwell, is recognized.
Thank you, Chair. Thank you to our panelists. People at home are terrified about Zika, and for good reason. And I've gone across the country with a group called Future Forum. It's 18 of the youngest members of our Democratic caucus, and we've engaged with thousands of millennials, young people who already are having financial difficulties starting a family, and now I think Zika, from what we've heard, is an additional just health difficulty that has been posed as far as when it comes to travel and planning for starting a family. So my first question, in light of that, does anyone on the panel disagree with the science behind the President's request for $1.9 billion in addressing this crisis? Okay, seeing that no one has answered, is anyone familiar with Ronald Klain's opinion piece in the Washington Post this weekend, "Zika is coming, but we're far from ready"? Has everyone read that piece? In that thoughtful piece, Mr. Klain points out that the funding approved in the House for the Ebola, taking money from the Ebola crisis to address the Zika crisis is like sending all the fire trucks from one city to help fight a blaze in another. It's short-sighted and dangerous. He also suggests that we should look at, to plan for future crises like this, having a public health emergency management agency. Any thoughts on any of the panelists as far as having a separate agency similar to FEMA but focused on public health emergencies? Yes, Dr. Presley.
I think one of the situations, and I don't know about California, but Texas is a home rule state. So every emergency is handled at the local level until other support is needed. And this is mainly to your last comments. Making another federal agency to funnel down money and hope that a little bit finally gets to the bottom on the ground, I do not think works. I think that's part of the problem now is we don't have the boots on the ground support that's needed.
Yes, Dr. Ernst.
I'd just like to echo that. I also think that, you know, maybe something within one of the existing agencies, for example, the Center for Disease Control, which has been leading a lot of the response in Puerto Rico, for example. But I also think trying to figure out a way where we can leverage other resources besides federal or state employees to respond would also be something that's useful. You know, every time that there's a crisis, there's a lot of volunteers, physicians and nurses and public health professionals who want to help. And we don't really have a good infrastructure in which they can be trained and deployed to help in these emergency situations. And so I think having some sort of infrastructure where people like myself and maybe some of the other panelists could use their expertise to help in the response would be really beneficial.
Great, thank you. And you know, my perspective is that people at home and across the country, they don't care whether it's a Republican solution or a Democratic solution that solves this. They just want to see government act in a crisis and get it solved. So thank you, Chairman. I yield back.
Thank you, Mr. Swalwell. And the gentlewoman from Maryland, Ms. Edwards, is recognized for her questions.
Thank you, Mr. Chairman, and thank you very much to the witnesses today. Really appreciate your testimony. Just a few weeks ago, I'm the co-chair of our Democratic Steering and Policy Committee. We held a hearing with Dr. Fauci, the Centers for Disease Control, the American Public Health Services, a mayor or so, about Zika. And one of the things that surprised me, because I didn't know very much, and looking at the maps, both the maps that we have here today, but also other maps sort of showing where the spheres of outbreaks are going to occur, it's pretty daunting. And especially if you look through the Gulf states, the South, and the Mid-Atlantic region. And so my question actually has to do with the spread, because although I understand there's a particular type of mosquito, I was surprised to know, and maybe I'm wrong about this, about whether the Aedes aegypti, is that how you pronounce it, mosquito, once it transmits to a human host, then can another mosquito, the traditionally found Asian tiger mosquito in the East, then transmit infected blood to another human being, or is that something that we know? Because to me, that suggests that we have just a much more potentially out of control circumstance. And I recognize there are a lot of things that we don't know, and this is tied to how we need to think about things like insecticide resistance, and whether we then have the capacity for the boots on the ground to have the resources that they need to be able to respond should there, which I presume there will be, a much wider spread or outbreak. And if anybody has any thoughts about that, I'd appreciate it.
Just on the transmission, if Aedes aegypti or Aedes albopictus, you mentioned the Asian tiger mosquito, it's not the blood, the infected blood that they're moving. Dr. Ernst had talked about extrinsic incubation, and that's once the mosquito picks up that virus has to move through the mosquito's body into the salivary glands where it can replicate the traditional model. And so there's a period of time, up to a week, more information is needed. So it's not like the mosquito can feed on the person and immediately go bite another person and be infective.
And so, but then the question gets that I have is, does it really matter what type of mosquito then?
Yes, it does. In this respect, Aedes aegypti, the way to think about it is Aedes aegypti is public enemy number one, responsible for far and away the greatest level of disease transmission, whether it's dengue or chikungunya, and now actually Zika. So Aedes albopictus is a species which is actually in its behavior, it's quite aggressive. It's a nasty biter, worse actually than aegypti. You notice it worse, the bite is worse. But actually it's a very inefficient disease vector. So in an ideal world, one would tackle both. But if you're looking at disease transmission and how to make an impact, tackle Aedes aegypti.
Got it. Then if you could talk again about the incubation period, because I think part of the testimony that we've heard both here and today is we don't know a lot about the incubation period. But there's a potential, especially if there's a transmission through sexual contact, that the incubation period could actually be much longer. So one might, for example, in terms of public health advice, advise a woman who's of childbearing age that there could potentially be a year and a half during which you really should not engage in sexual contact. Is that true?
Well, I think we don't really know exactly yet how long the virus can persist in semen. As Dr. Presley was saying, there has been some evidence up to 3 months after the infection was thought to occur. I would suggest that if possible, I know that the resources are really difficult to follow up each individual man until he's screens negative, but that's one potential possibility. The other possibility is that we do have some research that's ongoing that I have heard of, that they are following cohorts of men who are return travelers to really identify what is sort of that average time frame in which they can harbor the virus in their semen.
I guess, if there's 3 months and then add to that a 9-month pregnancy, that is a long window of time. And so I think it begs the question of how it is that we're advising people of childbearing age who've traveled to those regions what they need to think about. Anyway, I leave it at that. Thanks.
Thank you, Ms. Edwards. That concludes our hearing today. We thank you for your expert testimony. This has been one of the more interesting hearings that we've had. You all are experts in certain areas, and all of those areas were of interest to members of the committee today. So thank you all again. We may be back in touch, and I should also say that members have 2 weeks to submit questions to you all. You heard the comments today, how many questions went unanswered, so you may get some more questions in the next couple of weeks. Thank you all again, and we stand adjourned.